pleased to introduce Chris Munoz, who's a PhD candidate at the University of Texas at El Paso's Ecology and Evolutionary Biology program, where he works in Dr. Michael Moody's Plant Evolution Lab. Chris was highly recommended by Bill Norris and Russ, who's with us here tonight. So that they emailed me that they were very impressed with him and would we like to have a program? And of course I said yes. If you're on Zoom and you're not muted, please mute yourself. And if you have any questions or comments, please use the chat feature. Thanks, Chris.
Okay, thank you all for coming. My name is Chris. This is a talk on the evolution of a group of plants in a genus called Hebecarpa, belonging to the family Polygalaceae. And this is going to be all the latest and greatest of my PhD project. I'm in my 4th year. Before I continue, I do want to acknowledge my PI, Dr. Michael Moody, as well as the members of my committee, Drs. Darozet Nardi, Drs. LaRue, Harvey, and Mole as well. So to begin, there's a little delay on switching slides.
Sometimes they switch.
Well, they're not switching.
Use the mouse to put the cursor on the right screen, see if that helps.
I know it's not switching. Oh, on the right screen.
Yeah.
Maybe the down arrow will do it. Restart.
Honestly, just
Click.
switched 2 minutes ago.
We can restart. That's fine.
There we go.
There we go.
Just use the side arrow.
I was pressing it.
Sure.
I was pressing it. I promise.
Sure you were.
All right, do we start again?
Just keep going.
Okay, cool. All right, so to begin again, I want to go over the content of each of the 3 chapters that my project spans. So chapter 1 focuses on a phylogeny of the entire genus. And chapter 2 is going to be focusing on what we call genetic structure. So we're looking at population genomics across 2 species complexes called Obscura and Barbiana. More on that later. And then lastly, we're doing a character trait evolution analysis with a special emphasis on 2 structures, one called the elaiosome, which is actually pictured here on the bottom left, and another which is that of the overall morphology of the wings of the fruit. Okay, so the genus was first described by Abbott in 2009. He worked on this for his PhD. And essentially what happened is he ran a phylogenetic analysis and found that some of the taxa that were previously shoehorned into Polygala actually represent their own separate clades. And from that, other genera like Rhinotropis were born, so to speak. And since then, a paper by both Abbott and Pasteur came out in 2015 called the Preliminary Synopsis of Hebecarpa, where they essentially provided a list. There's a long list of 41 species that are morphologically diagnosable to Hebecarpa. And I'll explain what that means in just a second. So the taxonomic status, if you will, of this group is a little bit of a mess right now, just because a lot of work is forthcoming and pending. And though we think that there are 19 species, some workers assume that there are up to 70 species. So if you look at the photos on the top right, there's a photo of what is now called Seneca scoparioides. So by the way, all of the North American Polygala are now Seneca, okay? So there's a structure on the keel petal. So that keel-looking petal on the bottom in the middle has these finger-like appendages. Those are called fimbriate crests. Those are absent in true Hebecarpas, okay? So the list of 41 species that I just mentioned are diagnosable to Hebecarpa for that reason, among others, right? But that's one of the most diagnostic characteristics. So what I did as soon as I read that paper is I walked down to our herbarium at UTEP, University of Texas at El Paso. And essentially what I did was I checked to see what we had on that list in our herbarium, and we had Polygala brachycephala and Polygala magdalenae. So I extracted DNA from both. And we ran a PCR amplification on both of those, with only one of the two being successfully amplified. So what we did is we created a kind of preliminary analysis looking at true polygalas, again, now Seneca, but at the time this was Polygala alba, right? I included another genus called Monina, specifically the species Rhytii, and then some true Hebecarpas, right? And the idea is, if this list of 41 species in the case of Magdalene is representative of species that are truly diagnostic of Hebecarpas, then they should end up being nested within the Hebecarpa clade upon phylogenetic analysis. So this is a single gene analysis using the ITS region, but as you can see here, it ended up being nested within Hebecarpa. Polygala magdalene, by the way, is a species that's endemic to an island off of the coast of Baja California, an island called Isla Magdalena, right? So the name is simple enough. Okay, so before I move on to the next slide, there's one other thing I did want to point out, and this was kind of an accident. So we decided that we wanted to include, like I said, some Hebecarpas and some Polygalas. And so we threw in Hebecarpa barbiana from the Hueco Mountains and from the Capitan Mountains. And what we did not expect to see was divergence across those two populations. So you can see that there's divergence here between the Hueco Mountains population of Hebecarpa barbiana and the Capitan Mountains populations, which is interesting. It suggests that there's some kind of barrier to gene flow that perhaps has existed for enough time to promote divergence. So another one of the taxonomic mysteries of this group is one of the early workers named Blake. He was doing work in the early 20s. He identified, of course, based on morphology, various diagnostic characteristics of what he ended up delimiting into sections, right? And among these sections was a group called Adenophora. And Adenophora means gland bearing. And essentially what this is it's 3 species that are currently circumscribed under Hebecarpa. And those are Hebecarpa punctata on the top left from South Texas. It's a very rare plant from South Texas, where I'm from, actually. And then underneath that is Hebecarpa macrodinia. The third species is actually on the next slide. I'll kind of jump forward and back. And it's Hebecarpa evolvulacea. And this is a rare endemic to an area just east of San Luis Potosí in Mexico. Okay, so what we're looking at here is potentially a separate lineage. So we don't know if these traits are unique to essentially a separate clade. No one knows that because no one's done a phylogenetic analysis on this group among other Hebecarpa species. So we are doing that, and more on that shortly. But the last of the diagnostic structures that makes this group unique is not just the glands, but if you look at the flowers here, they have these lateral keel pouches, right? Those are absent in Hebecarpas. So this is Hebecarpa obscura from the Vix Peak area in central New Mexico. And then just like I was saying before we started recording, this is Hebecarpa obscura from the Huachuca Mountains in Arizona. Okay, so when we do phylogenetics, we make a lot of assumptions, right? And the models that we use are included among those assumptions. But to do something like time calibrated phylogeny, you have to tailor the analysis to the group that you're working on based on what you know about that group. So there's a paper that came out in 2019 by Pastor et al. And essentially it was this extremely inclusive, very well done phylogeny across Polygalaceae, and it included 3 species. Three species from each genus, right? Including Hebecarpa. And what we see is that the crown node of Hebecarpa, the most recent common ancestor of Hebecarpa right here, is dated to about 16.5 to 17 million years, right? And the way that they were able to calibrate this, if you're wondering, is they actually used a fossil. So when I say we use what we know about a group, we try to use everything that we could possibly make use of, right? So they used a calibration fossil called Paleoclimatic, and that's how they were able to time calibrate this ultrametric tree that they produced. So from this information, I was able to then include that as a component of our analysis. So into a little bit of the paleoclimatology that is pertinent to the group. So the first arrival indicated by that Pasteur phylogeny of the ancestor Hebecarpa into North America was about the Eocene, right? So about 50 million years ago. And why that should be confusing is primarily because the land masses of North and South America were disjunct at the time. They were not yet connected, okay? So how does a South American ancestor make its way across this body of water to another continent, right? So this is a mystery that a lot of people have been contending with for a long time, not just botanists. And one of the early hypotheses that gained a lot of momentum is the Garlandia hypothesis. So the GAAR in the name stands for Greater Antilles Aves Ridge, right? So it's this idea of like proto-islands that didn't necessarily connect North America to South America during the Eocene, but may have facilitated movement of biota between the two land masses, made it a little bit easier to kind of minimize the distance, right? Through island hopping. So this was a compelling idea, it still is, but some research has come out to challenge it. So in 2020, a paper by Roncall et al., which is a phylogenetics paper, found that about 80% of the fauna examined in the Caribbean islands had undergone adaptive radiations or diversification events after the Greater Antilles Aves Ridge became subaerial, right? Subaerial is just a way of saying it's under air and not underwater, right? So why that's relevant is it doesn't dismantle the hypothesis that they found this, but what it does is it kind of diminishes the importance of this as a corridor for dispersal, okay? So what really is a little bit more of a nail in the coffin, so to speak, is this paper from 2021, the following year, by Garak et al., where they did some geophysical analyses of the region, and they found that one of the more important tenets of the Garlandia hypothesis is not supported by geophysical evidence, at least in their study, and that is that the ridge, the islands, were not subaerial at the time that they were proposed to have been. So moving forward in time into the Miocene, which is, again, where we see that crown node of Hebecarpa at the 17 million year mark, because the Miocene ranges from about 21 million years to 5.3 million years, okay? So this is a time where the group that I work on is undergoing diversification based on the preliminary evidence that we have that you'll see shortly. So what was going on at the time? Well, major things were happening. First off, dominant assemblages of plants that were undergoing the C3 metabolic pathway had been replaced essentially very rapidly by C4 mediated photosynthesis. Now, how do we know that? I won't go into too much of it because when I did a timed run through of this talk, it was a little long the first time. So I'm going to keep it short. Essentially, people look at paleosols and they look at bioapatite or fossil tooth enamel, which is really neat. So if you look at this graph from Curling et al. In 1993, they looked at tooth enamel in fauna from Pakistan as well as North America. And what they're looking at specifically is because the metabolic pathways from C3 to C4 mediated photosynthesis are different, they sequester and they fix carbon in an inherently different way that results in a different isotopic signature, right? And that signature is recorded in the enamel and in soils that have enough biogenous material of the past, right? So that's what they did here. They looked at those signatures and how they changed over time, right? And what's compelling here is that this is a pattern that's being seen across the Northern Hemisphere, really, because they're looking at Pakistan and North America, and they also have some oceanic isotopic data to support it, because you can see on that vertical axis in millions of years how that drastic shift is happening at about the 8 million year mark to the 5 million year mark, okay? So you're going to want to remember that window of time going forward for this talk. Okay, so this is also a really important time for a lot of researchers because researchers love to debate things. They love to disagree. especially myself, right? So one of the things that's very hotly debated right now is when was the genesis of aridification of the American Southwest? Like when were our deserts born, so to speak, right? And the way that we tackle these kinds of questions in science is not through the means of one single discipline, right? We want to see that otherwise disparate approaches to understanding phenomena converge on the same narrative of history, right? So that paper that I mentioned by Garak et al. With the geophysics, that had implications for the Garlandia hypothesis, which is related to evolution and dispersal events. So similarly, this phylogeny, this is from a paper that came out in 2021 by Van Elst et al. On an ant genus. I'm not, I don't know a lot about animals, so wish me luck on this slide. But this is a genus where they essentially found a lot of cryptic diversity. It's a genus called Myrmecocystis. And what's really the important takeaway here is that there are these diversification events happening, right? So a single lineage is becoming multiple, and we're seeing that happen rather rapidly across this temporal scale, okay? And when are we seeing that happening? Well, we're seeing that happening at a time between about 15 and 5 million years ago, right? Arguably more in this paper between 10 and 5 million years ago. So a lot of speciation events are often facilitated by large scale displacement of previously dominant assemblages, whether it's plants or megafauna or whatever, right? So when climate shifts happen, there are cascading effects. I mean, we know that today, right? There are cascading effects on biota, and those are often reflected sometimes in the form of extinctions, sometimes in the form of adaptive radiations, like here. So this is kind of corroborating that something significant that affected biota was happening at about this time. And in particular, because this is a genus that is adapted to arid environments, that's even more of a suggestive finding. So what we do with phylogenies is we look at how species are related, right? We're interested in things like species delimitation, like in the ant paper, I noticed that there were cryptic species on the tips of those trees. We're interested in when splits occurred and the overall topology or the architecture of the tree so that we can understand what taxonomic revisions may be needed, right, for certain groups. But one of the things that we do in addition to that a lot of people don't really consider is we use phylogenies to inform other things, especially things related to paleoclimatology and proposed climate shifts. So one of the biggest challenges with the group that I work on is that it's comprised of a lot of really rare plants, which is what attracted me to the project to begin with, because that's kind of what got me into botany. It's like real life Pokemon hunting, except you're not like on your phone. So what I did was I contacted Tom Wendt, who's the former curator of the UT Austin Herbarium. Actually, no, I said that wrong. I contacted George Yatskovich, who's the current curator, and I wanted access to material to potentially extract DNA from it, right? And he then said, you'll have to talk to our designated curator of Polygalaceae. You may know him, Tom Wendt. And of course I do, because I've read a bunch of his work. So he actually worked on his PhD on a sister clade called Rhinotropis. Okay, so he's been very helpful. Essentially what he's doing is he's providing a lot of these specimens that I can't get to in Mexico for multiple reasons, right? They're often in very remote areas. Sometimes the areas are known to be dangerous, and this is just a better option. Alternatively, it's also nice to use these wonderful data consortia called herbaria, right? So when people go and they collect plants, they house these plants. The idea is that they get housed into perpetuity, right? You were talking about the unfortunate case of the Duke University Herbarium. Hopefully they get that sorted out, because what a loss that would be. These essentially house specimens that are extremely old, and all sorts of data can be derived from these consortia. So people look at morphological data. Many new species that haven't been described are just sitting in herbaria. And the other thing is, like me, they take tissue samples from these specimens not too much, just a little bit, so that we can make inferences about how things are related, especially in cases where getting fresh material is difficult or impossible. Because in some cases, they're extinct species that people are still able to derive DNA from because those species have gone extinct after they were collected. Hopefully the collector wasn't the reason. Right. So I've been working a lot with the people down at UT Austin, in the clock tower, actually. And the issue with that is that herbarium material is often old. So a lot of the specimens that I work with are collected, they were collected by Tom mostly in the 70s all over Mexico. And over time, DNA gets degraded, right? So I have been able, luckily, to obtain DNA from those specimens that he's given me, but not all of them, but some of them. And going forward, we'll have to see how much of a contribution that ends up being in terms of the types of phylogenetic differences we can make. So if you'll recall, the Pasteur phylogeny did what's called an ancestral range reconstruction. I was talking about the crown node of Hebecarpa, and you could see these pie charts on there. So those were showing the marginal probability of the tips of the ancestors of the taxa on the tips of that tree occupying a certain geographic space. So the way that we do this is we have sequence data, right? And then we create a matrix with notation indicating whether taxon is present or absent in a certain area that we designate as a region of interest, right? So that's something that I'm going to do with my data so that we can see not only when splits occurred temporally by making the tree ultrametric and time calibrating it, but also seeing where geographically these taxa were undergoing radiations, which should be extremely informative, especially when it comes to applying phylogenetics to addressing questions of paleoclimatology. So finally, some data, right? Okay, so we saw that single gene analysis. This is 29, I think it's 29 tips of taxa that is built from what's called ddRAD sequencing. So rather than using a single gene, ddRAD usually works with a range of about like 3,000 to 5,000 loci. So much more phylogenetically informative, much more resolute than a single gene analysis. And what we included here is a series of samples from Hebecarpa obscura in the San Mateo range of New Mexico near Vic's Peak. And then clade B is Hebecarpa barbiana from the Hueco Mountains, which is just east of El Paso. Then the teal clade is the Mule Mountains population of Hebecarpa obscura. And I'll touch on the green clade in just a second. So what's really an important takeaway here, there are a number of things, but the first thing I want to touch on is that these are currently circumscribed as the same species. And what this is showing us very clearly is that a taxonomic revision is needed. And I just finished about 2 weeks ago adding 96 samples to this ddRAD series of sequences. And we'll then do downstream analyses, and hopefully maybe another day I can do another talk and we'll see an even bigger tree, right? So the other thing that's interesting is that the closest related clade to Hebecarpa obscura here is that of what is currently circumscribed as a totally different species, barbiana. So just even more further underscoring the need for a taxonomic revision. Last two things. So if you'll recall that section of Denophora that Blake had outlined by looking at morphological characteristics, namely the pustular glands along the stems and leaves, and then the lateral keel pouches. So what I did is I included 2 out of the 3 of those species to see if they formed a separate clade. And this is the output. And you can see that they are diverged by about 17 million years. So pretty compelling evidence that this is probably in need of being treated as a separate genus entirely, with an entirely different evolutionary history. What I do want to do is I want to add Evolvulacea, that third species, to see if this topology is retained, right? As well as add more taxa within Hebecarpa. So the last thing on this phylogeny is that it's time calibrated, right? So the reason there's 2 here is that these are 2 different statistical methods that I used. One is a Bayesian method. If you have questions about that later, I'm more than happy to explain. And then the other one is a maximum likelihood method. And what we want to see, like I was saying before, is we want to see different methods converging on the same answers, right? And here we could see very similar topology with strong node support. indicating that there's a high degree of certainty that this is representative of what actually is happening taxonomically within the group. And the reason that that doesn't have all the tips kind of flattened out like this one does is because it's not ultrametric. It hasn't been time calibrated. And that's the last point I wanted to make here, is that if you look at the topology here, when are the majority of adaptive radiations and diversification events happening? Where are the splits happening? Well, they're happening in the last 5 million years, right? And we don't know if once I add those 96 samples, whether this will still look the same. But it is showing that at least for these taxa, that is the case. So these are somewhat recent radiations that coincided with these proposed shifts in climate and these increases in aridification in the Southwest. Okay, I promise chapter 2 and chapter 3 are shorter than chapter 1. So for chapter 2, we're also doing a phylogenetic analysis, but it's at a different scale, right? So here we're doing what's called population genomics. And we are using ddRad. In fact, that phylogeny I just showed you is a rad-seq phylogeny, right? But I threw it in just to give a little bit of insight with respect to Adenophora and the overall topology of the group. So what we're looking at here are two species. One is called Obscura, the other one's called Barbiana. And Obscura and Barbiana are widespread enough that we can collect them comprehensively, but they also have a very fragmented distribution, which is really ideal for studying patterns of speciation across time and understanding how geographic distributions might have shifted across time and will shift in the future. And I'll show you some models that I've created shortly on that. So what we're looking at here is these two species complexes, and we anticipate that there's cryptic speciation within these groups. So we just saw a little bit of that with the rad-seq data. And these are the sampling sites. So the red diamonds are Obscura populations. The blue diamonds are Barbiana. And the yellow one, Dr. Kleinman, the yellow one actually is something that Russ and I have been wondering about. It's rectipillus. So it's currently circumscribed as Hebecarpa rectipillus. And it was initially described as a separate species primarily because it has more dense vestiture of spreading trichomes rather than incurved trichomes along the stem. So species that are very similar to it are things like Barbiana and Obscura. And the question here that hopefully will be addressed by including it into the rad-seq analysis is it really a separate lineage? I don't know. It would be interesting if it was, but if it ends up being nested within one of these other groups, then we might have to lump that into either Barbiana or Obscura, or maybe even one of the new cryptic taxa that we describe as a result of this project. So I'm going to skip this slide just because I feel like that's maybe something better if you guys have questions about it later on. So when we try and understand gene flow phenomena by looking at high resolution scales, doing things like population genomics, we also need to understand what the barriers are to gene flow. Like, what is it that is preventing genes from being exchanged across populations? Is it distance alone? That's probably one of the more intuitive ideas that people have, and that's very common, right? Allopatric speciation happens all the time. But there are also other types of speciation, like sympatric speciation, where things are co-occurring, and yet there are more, let's say, ecological barriers to gene flow, right? Maybe some niche partitioning happens is happening within a relatively narrow space. So we want to understand what's happening within the group. So these are some preliminary SDMs that I made using some R scripts from, I think, Dr. Oliver I think he's at the University of Arizona. But essentially what we did here is we took 19 climate variables and we modeled where the habitat optima are across a continuum, right? So green representing the most optimal, and then deviations away from green ranging into the white, representing the least optimal habitat, right? And since this is an insular species, what I then did is I converted that spectrum to binary data so that habitat is not treated as a gradient of optimality, but rather either suitable or non-suitable, just to kind of visualize it a little bit easier so that I can make a projection about the distribution going forward. I plan to do this into the past as well, back at least to the last glacial maximum, like 20,000 years ago. But what you can see here is that for the upper panel, Obscura, the populations diminish pretty drastically going into the years 2061 and 2080. Less so for Barbiana, but you have to keep in mind that this is based on the current circumscription. And we already are seeing strong evidence that this really is multiple species. So if we then run the models again, treating them as such, then we can expect this to be a much more grim scenario. Sorry to bum y'all out. Okay, so there are only about 3 slides left, actually. So on chapter 3, we're looking at 2 traits. We're looking at the elastomes and the dimensions of the wings, okay? So human beings have an umbilical cord, right? So an embryo is attached to the ovary wall via an umbilical cord. Well, plants have something similar. So ovules are connected to the ovary wall via funiculus, right? And in some lineages, the funiculus is retained after fertilization and even into full fruit maturity and seed maturity. And so in this group, you could see this hat-like or this helmet-like structure is that retained aril. If you want a point of contact for maybe, or another example of what an aril looks like, if you've ever eaten a pomegranate, the part that is, I think everyone's favorite, the red part that's surrounding the seed, that's the aril actually that you're eating, right? So this is the aril in Hebecarpa. And we're interested in looking at how evolution has been shaped by adaptations within this group by focusing on adaptations that we think are structures that we think are related to aridity, right? Increased aridity. And the reason that we think that arils are important in that context is because of a paper by Lisci and let's see, I think it's actually on the next slide. Yeah. So a paper by Lisci and Pacini, where essentially they looked at the amount of water that was required in order to kickstart germination. And in those plants that had arils, the amount of water required was less. You can understand how it would be advantageous to require less of something that there's limited availability of in a desert, right? So the other structure is the wing, right? So the wing, it's pretty intuitive. The wing confers flight, and the wings are not the same throughout lineages. So from one species to the next, some wings are pretty big, some are very reduced. And we want to know whether there are selective pressures that are being imposed upon this group differentially across lineages, and whether that variability in wing dimensions is a result of those selective pressures being imposed. So one of the things that we did fairly early on is we looked at the 19 species of Hebecarpa that we were able to obtain measurements from, which is important to say, because I don't think every single one is represented here. I think there are maybe 1 or 2 missing. We also included a few of the Rhinotropus species here. So what we're looking at is degrees of habitat connectivity and the variation in wing dimensions, right? Because there was a hypothesis proposed, and it's discussed at length in Waters et al. 2020, where essentially taxa that are highly insular, taxa that are kind of confined to a very narrow niche space will, in some cases, in many cases, in fact, they will evolve a reduction in dispersal capabilities. Because if you think about it, let's say you're on an island of suitability and everything beyond a 10-foot buffer is not suitable habitat, well, then it will lead to the demise of that entity to disperse beyond that 10-foot buffer. So some lineages have reduced their ability to disperse as a result of being hyperinsular for long enough, right? So we were wondering if that was happening in this group. And in fact, the opposite was true, which sometimes happens in science. So what we did is we looked at everything that occurred north of the Trans-Mexican Volcanic Belt, because the habitat continuity is much more fragmented in that region, visibly much more fragmented just by looking at satellite imagery. And that's primarily due to changes in topography and geology. And what we found is that if the wings were bigger, that meant that the habitat connectivity was reduced, okay? So this was a very interesting finding, but the way that we went about collecting the data was rather simple, actually. There are a lot more elegant ways to measure habitat connectivity. And so going forward, we're gonna improve the methods that I used here. But that's it for this slide. And the last thing that I'll say pertains to a slide that I deleted, and that is that we want to see, once we have the full phylogeny of the genus, we want to see if diversification rates vary across lineages, and if those are constrained at all by these two traits, because that'll give us some insight as to how these groups have evolved with respect to things like adapting to an increasingly arid environment. And that's it. So I've got a lot of citations, but yeah, that's the talk. You guys have any questions?
See if there are already questions in the chat.
Sure.
Can I do? Okay.
Just when you're done with that, go back to that last slide.
Will do. Let me see. I don't see any questions in the chat.
Maybe.
Maybe.
Well, I can.
No, it's because of the cursor. Remember, I learned.
So when you talked about, there you go. There you go.
There.
When you talked about using the aerial, which I assume on that bottom picture is the yellow structure there at the top.
That is the arrow. That's the elizome. It's a carbuncular elizome.
The purpose was to use that as something that might help adapt to an arid environment, might help conserve water. At least that's what I remember you saying.
Yes.
But did that arrow initially cover the whole structure there? Because if it's just covering that little area, it's kind of hard to see how it's conserving much water.
Well, what happens is the arrow is packed with lipids, and during the seed's dormancy, it's using those lipids to maintain viability during that period of dormancy, right? And once there's enough moisture, the threshold for the moisture required to initiate germination is lower, which is advantageous in the presence of arrows than in the absence of arrows. So that's the idea.
And that doesn't happen, right?
What was that?
Does that happen in you use the pomegranate analogy, but
Oh, no, that doesn't, not that I know of, that doesn't happen in pomegranates.
Right.
I was just explaining that the structure that we call the funiculus or an aril is seen in pomegranates.
okay. So that would
Not that that's the explanation for what arils do across all lineages.
got it.
Yeah, that's within Polygalaceae.
Got it.
Yeah, specifically Polygalaceae. And then the other thing that I should add, since you asked about this, is that with respect to dispersal capabilities, there's another interesting paper, I believe it was by Forrest et al., and I think it's actually cited on the previous slide. Yeah, Forrest et al., 2007. So they found that that ants were preferentially collecting seeds with the arils intact and kind of ignoring those that didn't have arils.
So
And the idea
there's a vascular connection then between the aril and the seedlings.
Yeah, there is. Yeah, and in fact, removing it, because I've had to measure these and they're very, very small, is really hard. It's very well connected. And oftentimes I end up just losing them because they just pop off. But what I was going to say is that in the case of ant dispersal, before I saw this pattern that did not support my hypothesis about dispersal reduction, I was thinking that ants could actually be a form of dispersal reduction because the distances they travel are relatively short. And if you think about it, if you're from an evolutionary perspective, if you're already in a niche that's optimal for you, then it makes little sense to move other than to increase genetic variability of your species, right? So.
Yeah.
Yes.
In the Southwest, it usually happens, or it often happens that habitats with certain characteristics are widely separated. Like the riparian areas or the moist areas usually, are surrounded by total, non-moist area. And
Uh-huh.
you said that these were facultatively mesic, in other words,
Yeah,
they
so,
like a little bit of moisture.
yeah.
So,
mhm.
so do most of these that you've been studying look like the distribution was in the areas where they would be tended to be in isolated habitats?
Yeah, they have an affinity for desert margins, let's put it that way. And that's actually what makes this group so interesting. So studies have been done on Sky Island endemics, right? But at least within the Obscura and the Barbiana species complexes, you find these more at the ecotonal region between xeric habitat and characteristically mesic habitat. And that's a perfect situation, especially if you're doing ancestral range reconstruction for tracing the spatial extent geographically, right? And the way that the desert has moved over time, right? Because they are kind of occupying this niche of the desert's boundary, but they're not necessarily in mesic environments. I have found some Obscura populations in mesic environments. I just sequenced some, it was about 7 samples that are from some cliffs near the Cherry Creek campground up the road. Those look quite different, and you don't usually find them in places like that.
So they tend to outline the boundaries between the wet and dry.
Yes, exactly.
Ah.
Yeah. I think more than anything, it's that they're probably a relic of the Pleistocene. These are probably paleoendemics where maybe they had a previously more widespread distribution. And over time, as aridification increased, they had to kind of like follow that niche as it shifted across geographic space over time. And over time, it got more and more fragmented, of course.
You might want to check the.
So
Yeah, let me check.
seeing that, when you look at, they talked about the desert moving north like a kilometer a year or some such thing like that. With current climate change, are you seeing Hebecarpas moving north?
I did
Your
go,
lifetime?
so one of my biggest flexes is that I have really good absence data. Because I go and I look for these things and I don't find them a lot of the time. So for example, I went up to it was a stipulation of the permit that I wasn't supposed to talk about certain things, but I collected in southwestern Colorado. And well, I tried to collect because I had reason to believe that Obscura might be found there, based on niche modeling and things like that, as well as some questionable records from that area. And so I went and I didn't find it, right? But to answer your question, I do suspect that they do range farther north than they are reported, but I'm not sure that climate projections would lead them farther north. At least that's not what the models show. So.
Where are they? Both Baliana and Obscura are pretty rare around here, aren't they?
I wouldn't say that they're rare. Their distribution is very discontinuous. So it's very fragmented. So there are big gaps between populations, but there are a lot of populations. Again, some of those populations we have reason to believe should be treated as separate species. So in that context, then some of these populations are very rare.
What type of habitat around here would have they been? I think there was a spot more or less not in Silver City, but in our southwest area. What type of habitat would they be found? Would they be found in the burrows, for example?
In the burrows?
Yeah.
Where's that?
That's south of here. That's the mountains to the south.
Yeah, you can find Obscura just north of here. You can find Barbiana in every direction from here, essentially. But they're not very widespread. I would say, one of the things that you read a lot in species descriptions is locally abundant, and that certainly fits this group.
Is there an elevation requirement for these plants?
For Barbiana and Obscura, or for other species of Hebecarpa? Because the answer is yes for some of the narrow endemics of southern Mexico, like Colimensis, for instance. It's named after Colima, the volcano in Mexico. Yeah, so some of the species have an affinity, it seems an obligate affinity, in fact, to occur at high elevation. Obscura seems, I've seen Obscura at very high elevation in some of the sky islands in Arizona, which seems to be its center for diversity. And then I've seen it at very low elevations as well, just up the road. So. Because you guys are what, you said 7,000 feet here?
Six.
6? Yeah, so maybe by
Six
that
right
point.
here, and then you go up,
Mhm.
and Cherry Creek would be like seven.
Yeah,
Why is it called Obscura?
my advisor and I were at first thinking that it was because, just speculatively, we were thinking that it was because it's so hard to distinguish from Barbiana. But I think it has to do with the color of the fruit. Because it often has these really dark pigments. But I don't actually recall why it's called Obscura. I don't know why the epithet is Obscura. I read it once and decided to forget.
I noticed that most of the papers that you've cited are, to me at least, new, relatively new. Is this
Yeah.
whole area of research something that is becoming more popular or just?
Yeah. Applying phylogenetics to questions that are not directly related to the systematics of a group is becoming more and more popular. You're seeing across all of science, in fact, forget botany, across all of science, all of the kind of cutting edge findings are now integrative. A lot of progress is being made really by kind of forming these syncretisms between otherwise different disciplines. And that's definitely the case with phylogenetics. Like it used to be very compelling to put out a paper that's just on the systematics of a group. And it's becoming harder and harder to satisfy editors of journals with just that. Of course, there's journals that will take papers like that, but the more you're able to apply your phylogeny to bigger questions across fields, the more important it is to the scientific community. Yeah.
So then another question I have is, and I can't remember exactly what was showing up on a lot of your graphs. It has to do with the speed with which plants evolve and diversify. Does it happen maybe relatively quickly for whatever reason, and then slow down and take a long time, and then something else magical happens that we don't know about,
Yes.
and then speeds up again? And what kinds of things like that did you find?
So if you'll recall, going back to that DD-RAD phylogeny, which I'm not able to navigate to. So if you look at this phylogeny that's been time calibrated, you can see that situation that you're describing is what's happening here, right? And your question was, is that common? And I think another part of your question was, is it also common for diversification to happen at different rates across lineages?
Yeah.
Yes, that's very common. And that's actually one of the challenges of modern systematics, is trying to tailor models and calibrate models so that the amount of certainty that we have when we make these kinds of crazy inferences is optimized, right? And people, yeah, they do look at diversification rates across lineages, and that is something that we're doing. I believe that we're using BAM, but I don't recall the exact method that we're using. But you can see here that these are diversifying rather abruptly, right? And what's causing that, we don't know, right? That's a part of the mystery that we're trying to address.
Yeah, and I think that's a fascinating mystery.
Yeah, it
to
is.
solve.
It is. I mean, I think that what happened in this group is probably that you had a fairly widespread series of taxa geographically, and that over time, the climate became more arid, which which expanded habitat that was not suitable, right? And diminished optimal habitat and created isolation. And so therefore, I think that what's happening here is what's called geographical vicariance in the genetics community. Essentially, these are becoming more and more isolated. And as a byproduct of that isolation, they're no longer exchanging genes. Now you can do that. You can create a scenario like that today and then have a phylogeneticist look at that 100 years from now. Those would probably look like the same species, depending on mutation rates and other factors, right? To be seeing this level of divergence, and I don't have a table with the FST values. So the FST is genetic structure. It's the actual parameter that measures how genetically divergent taxa are, and in this context, populations are. And yeah, those numbers. So I remember one of the FST values for two populations of the same species in quotes was a greater FST value, meaning more genetic differentiation than a totally differently circumscribed species,
Wow.
which is a big red flag. And yeah, in a good way. Yeah, it means there's work to be done
More.
in terms of revisions.
Mhm.
Yeah.
It seems that you were saying that to get usable DNA from the various specimens, it has to be relatively recent usually.
Yeah.
But so people might say, well, what's the use of all the older ones? But the older ones can tell you, I guess, where something was at the time.
Oh, absolutely. Absolutely. Because ranges change. And sometimes, unfortunately, they change in our lifetime, often due to anthropogenic influence, right? So yeah, the metadata that we find on pressed specimens that end up being housed in herbaria is very useful for that exact reason. Because we can actually see not only how geographic ranges have changed, but even phenology. Like when things flower, right? Like you remember buying the seed packets like 30 years ago, right? So you buy a seed packet then and you compare it to one from now, like some of those have changed because the phenology has changed.
Don't get the feeling that the only reason we have herbaria is for DNA these days. Morphology is still important.
No, it's very important. No, it's.
I mean, there are reasons to have 150-year-old specimens out there.
Mhm.
Beyond just the DNA.
Absolutely. Even just my chapter 3, just the component of looking at habitat connectivity and the wing dimensions, I mean, that alone has nothing to do with genetics. That's more of a ecology question, yeah, that is then going to be applied to genetics. But
Wendy Graves here, and I just wanted to point out that a question was put into the chat shortly after you guys checked, so you might want to check again.
Let's see, any insects that use this genus as a host species? The answer is probably, but I don't know. If you are someone who can readily identify arthropods and things of that nature, I have photos that I've taken of various things that I'm very curious about that some of my entomology friends have not been able to identify. And maybe it's something new. So yeah, the answer is I don't know, but I've seen things on them and I have photos of that. Was that the only question in the chat? I think that's it. Yeah.
Can you go back to your Garlandia slide?
Oh, of course.
Slide.
Yeah, let me go back.
Maybe you can't.
I can.
There you go.
Ooh,
The one down there, that sure looks a lot like Central America bridging the, what do you call that part of Mexico that's sticking up there like with Cancun?
The Yucatan.
Yeah, it looks like Yucatan. And it looks like the Garlandia part is Central America leading down to South America.
Uh-huh.
And it has that, I mean, it doesn't take much imagination to see that there really,
Uh-huh.
right? So, I mean, if that's more or less the right way of looking at that, then there isn't a long distance between North America and Central America for that dispersal you were talking about. And it seems to me to be narrow-minded to think you have to have a land bridge at all. I mean,
It is.
birds and.
Mm-hmm. So yes, I agree. Just to be clear though, this is the configuration proposed at 34 million years ago, right? And the biggest thing here is that these islands were subaerial at this time. And this paper essentially demonstrated that that's not the case.
Oh,
Yeah, so there's that.
I mean, it doesn't look like they even needed to be there anyway.
Yeah, so based on the geophysical data, the Greater Antilles Abbas Ridge was not subaerial. So in other words, those islands were underwater at the time that they needed to be subaerial for the hypothesis to be what it is. The other point that you made is that it's narrow-minded to think that it has to be over land dispersal and that it can't be over water. Totally agree. And so did they actually in this paper. They specifically mention that over water dispersal mechanisms have to be taken more seriously, essentially. So one of the things I've been curious to know is like, well, it's hard to know what the ancestral states would have looked like for any given group, right?
There's plenty of
But-
reason to believe that, I mean, look at Ripsalis in Africa, right? The only active species that's native to Africa. It's also in South America.
And that is actually native,
It's native in South America, right? The genus is.
but you're saying it's found in Africa too? And that
It's
is,
the only cactus species.
I thought Cactaceae was endemic to North America. I didn't know that.
Yeah,
So it's mostly endemic.
Ripsalis in Africa.
Ripsalis. So what's the context for Ripsalis making its way to Africa in terms of proposed hypotheses?
I mean, the question has always been, I think it's been answered, but I must admit, I'm not a Ripsalis expert.
Okay, neither am I,
The
obviously.
question has always been, did that happen before or after the continents separated?
Oh, yeah.
But I think the answer to that was well after it has to be had with water dispersal. Does somebody know more about that than I do?
Didn't it-
Honestly, I'm not a cactus expert.
None of us here.
I used to write a wildlife column, and believe it or not, I wrote an article about the evolution of Cactaceae once, but this is like more than 10 years ago. And I'm remembering the number 40 million for the diversification of cacti from South America outward.
Well, there
But-
is one considered native to South Africa.
Interesting. So my point is it has to be recent, right? It
The
has,
problem is, we call it native there because it was there before humans, but
yeah.
humans haven't been there a heck of a long time.
Yeah, the last 8 million years, which is another reason why this kind of research applied to paleoclimatology of the Northern Hemisphere is important, because that's when our first lineages were undergoing evolution. Last 8 million years, 8 to 5 million. So, yeah.
Are the Hebecarpa insect pollinated?
Yes, I just don't know if they have specialists.
So,
Yeah, so that would be super interesting to look at.
I mean, wind pollinated, you could, would spread easier, but if they're insect pollinated, depends on what kind of insects and how far they can fly, if they can go over water.
Yeah. Time is such a currency, right? Yeah.
So let me ask you.
When
Oh, go ahead.
I was just going to point out that there is another comment or another question in the chat.
Okay, thank you. Let me look. Let's see, I think it's another one from John. John, nice to meet you. Yeah, I would love to see the photos. Okay, and then your email, thank you. And do you use iNaturalist? And if so, what is your handle? I feel like this is a very divisive thing to say, but I don't use iNaturalist. I think it's awesome. And I feel like I advocate for it a lot, like for someone who doesn't use it. It's super useful, obviously. I sometimes look at the observations on my group in Mexico. So I guess to that extent, I do use it, but I don't actually have a profile on there. I'm sorry to say. Yeah. Wasn't that devised by a master's student at Berkeley or Stanford?
I don't know.
What an amazing master's project, right?
Yeah.
If that's the case.
So the first time, second time, third time, fourth time, I saw Hebecarpa, it's called Polygalaceae.
Yeah.
I looked at it and trying to figure out what it was, and it sure looked like something with heavies.
It's in the Fabales, so that makes
Well,
sense.
that's what I was gonna ask. It took me forever, literally forever. And I think I had to ask Dr. Zimmer. I didn't remember right. what the heck this darn thing was. Because
Yeah.
I didn't key out the Fabaceae or darn. My question was gonna be, it must be closely related to Fabaceae.
Yeah, it's in the order Phibales. So the first Hebecarpa that I saw was before I was cured of plant blindness, of course. I'm sure I've walked by them countless times. But when I started getting into botany, I was on a friend's ranch and I found 3 county records while on that ranch. And one of them was this plant I couldn't identify. And I'm like, what is this bean? Like, I was so overly certain that it was Fabaceae, right? And I never knew I was going to work on this so much.
You're in the same boat I was in, but
Yeah, exactly. And I remember it was just this yellow flowered looking bean that was growing in calcium carbonate rocks in a creek on private property. And eventually I found it. And yeah, it was Hebecarpa ovatifolia. And that was near Ozona and like Junction,
oh, yeah.
that area, kind of the hill country. Yeah. But yeah, they have a lot of morphological attributes that look like the papillonoideae, right? They have like the sepals on the side that are called wing petals, but they're actually sepals. And they have the keel petal as well. So. I don't think I have anything else in the chat.
Thank you very much.
Yes.
Yeah, of course. My pleasure.
For coming over.
Yeah, of course.
So what were you doing in Boone County, Texas? Where-
It wasn't Boone. It was, what was it? It was,
Boone County?
I'm trying to remember. Those are just like points on the map that are the closest. He's like way out in the boonies. No relation to Boone. No relation to Boone.
Ozona is the capital city for some small county.
Rock Springs. That's the closest town to where he was at. Yeah, Rock Springs.